All right, so now we're going to keep going and finish up this part on genetics. So we've been focusing on genes that are just located on the autosomes. So for us, it's chromosomes 1 through 22. But you do have that X and the Y, which are your sex chromosomes. So what we're going to do is talk about what happens when genes are located on there. We did talk about one gene, which was the SRY gene, located on the Y chromosome. And there are other genes that are too. There are genes affecting just normal maintenance of the body. So you have to, especially on X chromosome, so you at least have to have at least one X. And most of the time people, when they think of sex chromosomes, they just affect fertility. And it is true, there are a lot of genes that affect fertility on the X and the Y, but there are some genes just for normal maintenance of the body on the X. So if you don't have an X, the embryo pretty much will be terminated. It's not going to be functional. Now, when it comes to us, if we have the combination of XX, we wind up being female, XY, you're male. Now, I showed you when I talked about chromosomes, you do get extra copies of the X quite often. A lot of this genetic disorders happen when non-disjunction happens. You have multiple X's. And one thing you might have seen, if you go check out that tables, you see a lot of developmental problems happen. Now, if you ever had seen families where it seems like they only have girls or only have boys, it all comes down to dad. Mom's only going to contribute an X, so mom gives it that. If you're going to determine if you're going to be male or female, it all comes down to dad and how the sperm created. So maybe his X swimmers are a little more faster than his Y swimmers if he just has daughters, or if it's a family that only has boys, those Y sperm kind of outperform the X ones. It's kind of interesting. You see these kind of things happen in families. In my family, my dad only had daughters. Then we go to the next generation on my mom's side for all the great grandkids, and they've all been boys except for one girl. It's kind of crazy how things happen. So when we're talking about sex-linked genes, the one I'm going to focus on is color blindness. This is probably the most common one that people think about. Now, why are you barking? Dog's watch, sitting here with me for this one. So the genes can be located on the X or the Y. We're going to see most of them on the X, and we're going to focus on color blindness because it is located on the X. Now with females, and color blindness is a recessive gene, so in order to be colorblind, you have to have the recessive allele. Now for males, you have a little bit of a disadvantage of this. Since you only have one X, you're either going to get the normal allele or the allele that causes color blindness. But for females, we have the alternatives where we have the two X's. So if we have one allele that has the normal dominant allele and one that has a recessive one that causes colorblind, she's heterozygous. She's going to be what we call a female. She is a female, but she's going to be called a carrier. By that means she has a normal phenotype, but her genotype has a recessive allele that can be passed on. Now when this happens, so this is an example, we have a male with normal vision and a female that's a carrier. This will determine how everything is split up between the kids. So you can have, with this example, we're going to have an unaffected son and an unaffected daughter. They're going to have normal vision. And then we're going to have this other daughter. She's going to have normal vision too, but she's also going to be a carrier because mom's going to give her allele that has the gene for color blindness. But then we have this kid right here. Now what happens is dad gave him his Y, but mom gave him her allele with the bad gene on it, the X, so he's going to be colorblind. I'm not saying that colorblind is not bad. My brother-in-law is colorblind, and we have a lot of fun with him, and and I know quite a few people. It's actually kind of fun. It's kind of interesting. One thing about this, since females, we can be the carriers, this is why you see more men than female that are colorblind, and it's really cool. So this is a good little legend for you because it writes out recessive disorders on the X chromosome. So for males, these are the two different genotypes you can have. So they're going to have the dominant allele, they'll have no disorder, or the recessive allele, they will have disorder. It's either or, what you get. Now females, we have these three combinations we can be. We can be homozygous dominant, so we'll have normal vision, heterozygous, to where we have one normal allele, one recessive, and you'll still have normal vision, or you can be where they're both recessive, and this is where they will be colorblind. So this is just working through like a cross right here. Now let's work through one. So I'm not going to write out the legend because it's on the previous slide and you can go back and look it, but we're going to use what we know to solve this. And with these, most of the time you'll do letters with superscripts. I really can't type that and collaborate, so I'm just going to put the letter right next to it and then a space. But one thing about sex-linked crosses, when you do these Punnett squares, it tells you both the gender as well as the genotype, and you can then give the phenotype. So pay attention when the questions, when you're reading them, because I will ask you about gender in them. So be very particular. I've noticed on previous assignments, students kind of overlook that and they just count everyone in general. So we have a colorblind woman and a man with normal vision start a family. So I'm going to type in right here. So we know mom, she is colorblind. So you have to put the X's on it. So X little r, X little r. She's colorblind. The only way that she can be colorblind is if she has both recessive alleles. So we know mom is this. And then a man with normal vision. So he is going to be X big R, Y. Okay. So remember these Y's should be superscript, but it's kind of impossible to do on collaborate. This is when I really missed my whiteboard and like the mistake I did on the Punnett square one. So now what we're going to do is split out the gametes. So we're going to have one of our X's over here, and here's our other X over here. And we'll split out dad, he's a big R, and then this is his Y. So how the gametes split out, here's one gamete. Boom, there's one egg. Here's her other egg. Boom. Here's one sperm. Here's your little swimmer. And here's the other one. Goes right here. So remember the R, it's not a different trait. It's associated with the X. Normally it's like up in a superscript. So now let's go through and complete like you did a normal monohybrid. So let's pick R. Right, so I'm going to go through and type each of these in and make sure I do not push clear. What? Okay. So one thing I want to point out is you find out both the gender of the offspring and also the genotype, which will tell you the phenotype. So let's read our question. What percent of the sons would be expected to have normal vision? So this is where you can go to the previous slide where it has the table, the legend on it. So you know that it's a dominant trait, and I want to know sons that have normal vision. So they have to have the dominant R. So we're going to look for the sons, which is these two right here. You look at them both. Okay, they both have the little r. Both of the kids, the boys, are colorblind. So you can say big old goose egg right there, 0%. Now your next question, what percent of the daughters, so you're focusing on girls only, would be expected to be colorblind? So we're looking at our girls right here. And we look at their genotypic combinations, and they're both have that dominant R. So that means they're going to have normal vision. So you get, they're both going to have normal vision, but they are carriers. All right, and then you can put like both are carriers. Both are carriers. That's a cool little thing right there. I might ask you what percentage are carriers. All right, so pay attention when you're talking about genders. You're just talking about 50%, 25%, not out of 4, just out of the gender. Now, as girls with those 2 X's come some problems. Not bad ones, but we just have to compensate. Now, with the extra X, one thing we got to make sure is we don't have an excess of gene dosage, because the X will make RNA, then that makes more protein, and then we can have some abnormal levels. So what we do to combat this is a process called X inactivation. So what that means is randomly during development, this is done, and in every cell, an X is selected. And what happens is that X is kind of smushed down and made into called a bar body. Now by that, it's just kind of compressed down. Now it's random how it's decided, regardless of what the genotype or which allele that X have, doesn't matter if it's going to be inactivated or not. Calico cats are a prime example. So if you have a little orange patch right here on her cheek, what happened in that patch of cells right there, the allele for hair color that was orange is remained active, and all X's that in each cell that had the allele for black hair were inactivated and compressed into that bar body. So if we find a black patch like on her tail, the X alleles were the alleles with the orange hair color were compacted into the bar body, and then the ones with the black allele were active. And that leads to this mosaic pattern that you will see. Now, we do break the rules. We've been talking about complete dominance so far, and we know, like, okay, across this, I expect this ratio to happen. The more you Punnett squares, you'll get these ratios memorized. Now, with Mendel's work, the more and more we figured out, we've learned all this math with it and all the chi-square analysis, and it gets a lot more connected. But we started to see sometimes where the math didn't add up. We didn't get these classic 3-to-1 ratios or the 9 to 3 to 3-to-1. So what that indicates is, okay, things not right. Well, that may lead to where we have some different patterns of inheritance. So we've been talking about complete dominance so far. By complete dominance, I was telling you that the dominant allele is the complete boss. And anytime you had a heterozygote, it's had the same phenotype as the dominant. But there's also there's instances where this rule is broken. So one example is called incomplete dominance. So this is like, I call it like a mixing of the paint. So what happens is the heterozygote doesn't display either of the phenotypes of the parents. It's going to have the genotype, but it's going to have different phenotypes. So it's not like it's this or that, it's going to be a combination of both. So it's like if you were mixing paint. So let's use this top one to explain that. So we have red color, which is dominant to white color in flowers. So if I have a true breeding red plant crossed with the white true breeder, all of the offspring are going to be heterozygous. But when it comes to their phenotype, they're pink. Kind of looks purple here. But what happens is, like, if you took both of these and kind of mixed them together and you got this pinkish color. So the heterozygote falls in the range between the two of them. Another one is codominance. So with codominance, it's not like mixing the paint, it's like taking patches and making kind of a patchwork quilt of both of the parents. The heterozygote, it's not an intermediate, it actually shows both of the phenotypes. So I know this is a cute little cartoon. It's not how panda bears are made, but it's kind of cute, and we're going to talk about it in blood typing. So if I took a white bear and a black bear, and they had a great time and they made babies, this is showing what codominance is. They're not gray, like it was if it's complete, but we see parts of both parents. So here's the white phenotype of the face, and then dad's phenotype of his feet and his ears are showing up. So let's relate codominance to blood types. So we're talking about the antigens on your cells. Now there's 4 different blood types. We got type A, type B, AB, and O. Now we use this little I right here, and then the antigen that you have, or lack of, is shown right here. So if you have the antigen for A and your type A blood, you will have this combination. So if you can be homozygous or heterozygous right here. So this is showing you the blood type, and here's the genotype, and blood type is more like the phenotype. So you could be homozygous dominant, have both of the A antigens, or you could be heterozygous and have one A antigen and then not have another one. So that would be type O. For type B antigen, all right, so you could be homozygous or heterozygous. And then type AB, this is a codominant one. So you will have both the antigens for A and B. So one allele is A, one is going to be B. Both are showing up. They're both equally expressed. And then if type O, you got nothing. Sorry about you. But people really want your blood type because it's kind of great. So what we can do from this is do like several like little problems. And blood typing games are so much fun to play in class. I wish we could have done it. And you'll have like one problem on the homework that you're going to go through and do it. So let's look at one. Or actually look at two. So we're looking at these two problems right here. We'll do this first one. And this is one, it's kind of fun. It makes you work backwards. So pose a child of blood type A, so we know a kid's type of blood, and mom is type O. It's asking you what type or types might dad be. So let me see if I can type out my legend here. I'm going to do lowercase just to make it a little bit easier for us. So let me fix this. Let's see, I'm going to use my eraser here the right way. So let me type it here. I-A-R-I-A-I-O equals type A. It looks like Lala, it really does. But that's the font we have, so we will go with it. I-B, I-B-R, I-B-I-O. I promise those are I's right there. That's going to be B. Let's see. I-A, I-B. It's going to be type AB. And then you could be I-O, I-O. And that would be type O. Not type 0, type O. All right, so this is a little legend. So we know the kid is type A and mom is type O. So for this one, I'm gonna write it out with the pencil, so bear with me here. We know the kid has to be type A. So he's gonna be either this or this. And mom is type O, so that's telling you that he has to be this because he got one of his alleles from mom. So his other allele happens to be, boom, this right here. Now what we need to do is determine what's going to happen. What could a dad been? So we can do some reasoning right here. If we go through, we know that mom gave him, we'll put mom's allele right here. All right, and then we know this other one is dad's allele. Okay. So we know mom gave him this combination. Dad had to give us. So now what we need to go through and see all different combinations that dad possibly can be. So dad could be type A. He could have been either of these genotypes, so he could be type A. There's no way dad could be type B because there's no IA there. Dad could be type AB. If you look at that right there. So if you look at it right here, if dad would have passed this IA right here, that would work. No way that dad could be type O. So the blood types that dad could be are type A and type AB. So this is just kind of working backwards to solve a problem. You can do all the different Punnett squares, fill it in for each of them to see what you get. So let's look at this next one. Suppose a father of blood type A and a mother of blood type B have a type O child. Okay, what are the blood types of the possible future children? Let me get them all top over here. So you know they had a type O kid, so that tells you that they are this combination are right here. You know they have to be this combination because they had a type O kid, so they had both of them had to have that type O allele. What you would do now is kind of put it into your Punnett square. So we'll do mom or whoever has type A here. Let's see. And this is another way you can also use the subscript to make it a lot easier to understand, and it doesn't look like pounds and okay. So now we'll just go through and do our Punnett square. Okay. So let me get back to that pointer. So looking at this, these two parents, they could have a kid that's type AB blood. They can have a kid that's just type B blood. They can have a kid that's type A blood, or they can have a kid that's type O. I know there's positive, negatives. We're keeping it super simple with here, but they can have the whole smorgasbord with these. There's one problem that you'll answer on your homework about blood typing. Now, another thing that you can do in genetics that's kind of fun is pedigrees. So this is just a pictorial representation of family history. Some people love to do this. They can go through and track all the different generations. Each line is a specific generation. So here's like two parents, and then these are their children. You mark the males by squares, females by circles, and then whatever trait you're looking on will determine if you're going to leave it solid or if you're going to leave it blank. And these are actually kind of fun because with these you can actually look for genetic problems. So you can actually determine if traits are autosomal or if they're sex linked or if they're dominant or recessive. They're like kind of fun. When I teach genetics, we actually do some of these problems because, you know, it's like a detective and you're looking for certain patterns. So these are fun. I've done them before in class where we'd actually do a project where we actually go through and talk to your family and see if there's different traits. You could do it like with the left thumb, right thumb when you put your hands together, or you know, if your second toe is longer than your big toe. There's all kinds of weird things you can do. It's actually kind of fun to do. So genetics and ethics go hand in hand. It's actually really fun to talk about because when it comes to genetic testing, it makes us question, should we be doing it? Because we can get all this information, but why do we need it all? You know, I put some key points right here. So genetic information often identifies risk of medical conditions, and that might not be affecting you. So, you know, sometimes that's good to know if you're at risk, and maybe, you know, you can do some preventive things to risk it from oncoming. But what if it's a condition you can't stop no matter what? So that's kind of scary. It can also be about families as well as individuals. So what if you have found out something in a certain condition, and then it might scare if you have siblings and they have to go and get tested to see if it works out that way too. And then, you know, will your parents feel guilty for passing it on to you? Or maybe it's, you know, it's a recessive disease and, you know, they were both heterozygotes. You know, it's kind of interesting. It could fit about the families. It's not just you. And this is it's commercially driven right now. When you spit in a cup and send it off for ancestry, they own your DNA now, and can that be used against you? So why would you do genetic testing? Well, sometimes you see there's a condition. We went through this with my father. He was diagnosed with progressive supranuclear palsy, and I lost him about 10 years ago. And one of the first things they asked was, did I pass this on to my kids? Is it a genetic condition? And it's not, but I can understand wanting to know that because I've seen what it does to the body. I would be completely terrified and I don't know what I would do. But you will go through this and you see a condition, you'll do all this stuff and you'll try to figure out the cause. You know, if you know it's caused by chromosomes, or maybe it's just something that happened thanks to mutations, exposure to carcinogens, try to figure out the cause. Now, sometimes you'll have to meet with the family and explain it is. And you might have, if it's a genetic condition, you might conduct a family pedigree. And you might have to go through this thing. What's the probability of transmitting to future generations? So that might lead to conversations about reproductive options. You know, is it ethically right for you to have conditions knowing that you're going to give them this possible condition? You know, what if it's a recessive condition that there's no cure for, but you're both heterozygous for it? So there's a 25% chance a child is going to get both recessive alleles. You know, is that right to take the risk? It kind of makes you wonder. Now, when it comes to deciding this, there's like so many factors that will make people make that decision. And, you know, this is something we would have talked about in class because, you know, everyone has different views. I always think of people like icebergs. You see the top part, but you don't know about all that's underwater that led to their views and all the other stuff. So, you know, respect what people make a decision for a reason. You don't know everything. Now, this field of genetic testing is really taken off, especially with, you know, the at-home kits. You can spit in a cup, send it off, and they send you a bunch of information. Most people just do it for the ancestry, but what if you find out some of these traits and health conditions? Do you know how to understand the information? Are we giving people too much information? Because yeah, it's great that you find out all this stuff about you, but what if you find out something bad? Do you know how to comprehend it? So there are people that are going through these programs from genetic counselors. I know someone who's doing it, and I find it quite interesting because you're helping people understand, this is what you have, and this is how we understand what's going on. Now, there are risks for getting tested. Two big ones that I have right here. One is the psychological stuff. So what happens if you find out you're positive for a bad mutation? I know it's kind of there, but it's a certain genetic disease. And what if it's something that does come on? For example, it'd be Huntington's. It might not onset until you're later on, maybe in your 50s and you're in your 20s. And, it's shown to run in your family, blah, blah, blah. So you get tested and you find out you're positive. I've seen it go both ways. Some people are planners. They'd want to do and plan as much as life and do everything they can before they can possibly start to set in. I've seen people that are like, they just don't want to know. If it happens, it happens, and I'm going to live my life no matter what. Or what happens if you get the test and you find out you're negative? It's like, I call it survivor guilt, breast cancer. So, we know it is genetic, and maybe you wound up having the good versions of the alleles, but then your sister had the bad ones. So it makes you think, it's like, okay, is that good or bad? It's like, oh, I got lucky, but not you. So, you have to think about that. And then the practical part, can this be used against you? When you submit this, can your employer find out that, oh, well, in about 10 years, they're going to have this condition? Would insurance be affected? Or health insurance? That's a big one because we can't figure it out. Could the police or government possibly use your DNA to help solve crimes? And maybe, it's like, oh, here's your sequence, and we have all this untold stuff, and we just matched your sequence to this crime scene a long time ago, and boom. And there's enough similarities between your DNA and distant relatives, so even like distant relatives similar enough can get you kind of caught. And it makes you wonder, when you submit this information, what can they do with it? So it makes you really wonder. So here are some more genetic review videos on the Punnett Square assignment. There are a lot of them on there too. It might be some of the same ones. But don't be afraid to ask help. If any of these topics interest you and you want to talk about it, those are what those online sessions are really good for, or we can set one up. So I'm going to stop it right there.